Hi everyone. Today we are going to discuss some topics from blood from the chapter Body Fluids and Circulation. We all know that living cells need to be provided with nutrients, oxygen and other essential substances. And also in the same way, toxic and harmful substances need to be removed continuously for healthy functioning of tissues. Therefore, it is essential to have efficient mechanisms for the movement of these substances to the cells and from the cells. There are two pictures. One is for sponges, another is for coelenterata. Sponges come under porifera. Different animals have obtained different methods. First of all, we will discuss the types of transportation, and these types of transportation is based on the medium. Type 1, where water is the medium. And the examples I have shown you in the last slide, that is the sponges, water canal system. Hydra, they use the gastrovascular system. Starfish from Echinodermata, they use the ambulacral or water vascular system. The second type is where the blood is the medium. Again, in type 2, where the blood is the medium, there are two types: open circulatory system and closed circulatory system. In open circulatory system, the blood flows in vessels opening into the tissue spaces or membrane sinuses. And the examples is arthropods and most mollusks. And what happens in closed circulatory system is the blood flows through heart and through certain vessels and capillaries without coming in direct contact with lined sinuses or the body cavity. To be noted that blood was first discovered in the year 1628 by William Harvey. So let's highlight this point. Now we are going to discuss on the blood. Blood is actually a specialized connective tissue. Blood contains plasma and another is formed elements. The plasma constitutes 55% and the formed elements, it constitutes 45%. You can see here the elements of blood. The plasma makes 55%, platelets makes 0.01%, red blood cells it makes about 41%, white blood cells for 4%. And under white blood cells we have lymphocyte, basophils, eosinophil, monocyte and neutrophil. We'll be discussing this in detail. So now we are going to discuss on the plasma. The plasma is a straw-colored viscous fluid forming the matrix of the blood. So what is a matrix? Matrix is the ground substance of a tissue. It is generally a non-living substance occupying the space between cells. Plasma is made up of water. Water means 90 to 92%. And proteins, it makes 6 to 8%. So what are the different proteins which are found in plasma? Let us see. The different proteins are the fibrinogen, globulins and albumins. The fibrinogen is actually the clotting factor which is present in your blood. Globulins, this helps in defense mechanism. This can be subdivided into the alpha, beta and gamma types. Remember, this alpha and beta, these are lipid soluble and helps in vitamin transportation, whereas the gamma produces antibodies and helps in defense mechanism. The albumins sustain blood volume and pressure. Another point to be noted is that if you take a whole blood and if you undergo centrifugation, then you will see that the erythrocytes, which constitutes 45% of total blood, settles down, and the plasma 55% of total blood, that is on the upper surface of the erythrocytes. In the middle of the plasma and erythrocytes, there is a buffy coat which contains leukocytes and platelets. Leukocytes means the WBCs and platelets, and it constitutes less than 1% of total blood. Moving to the next slide, we can see that plasma also contains minerals like sodium, calcium, magnesium, bicarbonate ions, chloride ions. Other than this, there are glucose, amino acids, lipids. These are also found in plasma. And to be remembered that plasma without clotting proteins is called serum. So here I have made a note. Just remember that plasma is actually blood without blood cells. I mean blood without the formed elements. That is called plasma. And the serum is actually plasma without clotting factor. And the clotting factor or the clotting protein is the fibrinogen. So plasma without fibrinogen is called the serum. Highlight this point that plasma is blood without blood cells and serum is plasma without fibrinogen. This is very important, just remember. Now we are going to have a detailed discussion about the formed elements. So in this lecture I will be talking up to the RBCs, and in the coming lectures I will be discussing about WBCs and platelets. So we are starting with the first formed element, that is the erythrocytes or the red blood cell. What are the common features of erythrocytes? Erythrocytes are enucleated. We will not call it that it is non-nucleated, it is enucleated, because in the earlier stages the RBCs have nucleus, but while the RBC matures, that time there is no nucleus, so we will call it enucleated. So the RBCs are enucleated, biconcave, and it is discoidal. To be mentioned here that these RBCs are biconcave and discoidal to accommodate more hemoglobin. Mitochondria is absent. It is formed in red bone marrow. It is destroyed in spleen, which is also the graveyard of RBC. And the lifespan of RBC or erythrocyte is 120 days. That is 4 months. Here you can see the pictures that it is actually biconcave in shape, discoidal, you can see here. This is to facilitate the accommodation of more amount of hemoglobin. So now we will discuss the count of erythrocytes. The normal count of erythrocytes in males is 5 to 5.5 million per millimeter cube, and the normal count in females is 4.5 to 5 million per millimeter cube. This is the normal count. However, in some people, it is found that there is an increase in RBC count. This condition we will call it as polycythemia, and when there is decrease in RBC count, we will call it as erythrocytopenia. To be noted that one hemoglobin molecule contains 4 O2 molecules, and one RBC contains 250 million moles of hemoglobin. And also remember that normal hemoglobin level in any body is 12 to 16 gram per ml. Again, here a disease is involved which is called anemia. Generally, we get confused between erythrocytopenia and anemia. But remember, when there is decrease in hemoglobin level, it is called anemia. And when there is decrease in RBC count, it is called erythrocytopenia. So these two are totally different. And the process of formation of RBC or the erythrocytes is called erythropoiesis. You can see the difference between a normal hand and anemic person. Anemic person generally their hands, their skin gets pale, and this is how it looks. Now moving to the next slide. Now the last slide, we will discuss the functions of RBC. We all know what are the functions of RBC. First of all, RBC helps in the transport of oxygen in the form of oxyhemoglobin, and it also helps in the transportation of carbon dioxide in the form of carboaminohemoglobin. It also helps in the conversion of carbon dioxide into bicarbonate in presence of enzyme known as carbonic anhydrase. Remember this point, this is very important. Conversion takes place in the presence of enzyme called carbonic anhydrase. Now we will discuss the most important point, that what are the sites of erythropoiesis or the formation of RBC. During the early embryonic stage, the site of erythropoiesis is the yolk sac. During the late embryonic stage, I mean the last 2 to 3 months of intrauterine life, it occurs in the liver and spleen. And after birth, the site of erythropoiesis, as I have discussed, it is the red bone marrow. Again, I will say that remember it's red bone marrow, not just bone marrow, because in body we have two types of bone marrow, red bone marrow and yellow bone marrow. So it's RBM, and YBM functions in special cases. So this was all about the erythrocytes. In the coming discussions or coming lectures, we will discuss about the WBCs and platelets. So I hope this is clear. Don't forget to like, share and comment, and press on the bell icon to subscribe now. Thank you.